Hello, once more. Good afternoon, my colleagues, my students. I
Good
welcome
morning,
you
sir.
all. Yes,
Good afternoon, sir.
afternoon. How are you?
Fine, sir. Fine, thank you, sir. Fine, sir.
Okay, thank you very. Yes, yeah. Welcome to today's facilitation.
Thank you,
Yeah.
sir.
Yes, thank you very much. I hope you people are fine.
Yes, sir.
Yes, I got some of your assignments. Some I don't know. Some have not complete sent, but those that I've sent, I'm possession of them, and I'll do the needful. So today we are going to continue from where we stopped last week. So last week we looked about the living things and non-living things, and then from there we now went to the cell, and then we looked at the cell, the structure of the cell, cells in plants and cells in animals. And then we draw and label plant cell, animal cells. And then we look at the differences between them. We look at similarities. From there, we now looked at we generally looked at the cell theory, and then look at scientists that contributed to the development of cell theory. That was what we did last week. So today we are going to continue from where we stopped. So we are going to look at cell division today. So today we are going to look at cell division. Cell, as you know, is the basic structural and functional unit of life. And then division means a kind of separating. So the division that takes place in a cell is called cell division. And so the cells in plants divide, and also the cells in animals divide. For the organism to grow, the cells must divide. Remember when you were born, you are very tender, but at the age 1 month old, there's a difference. 1 year old, there's a difference. So your cells are dividing, bringing about the growth and development of the individual. So division takes place in animals, and also division takes place in plants, plant cells and animal cells. So let's now go to so once division stops, then the animals stop growing, and problem will occur. If you have a cut in your body, a time will now come when that place heals. And so it's the cells that are dividing and dividing, making sure that your body that has an injury is healed. So let's go to look at types of cell division. Types of cell division. So we are going to look at types of cell division. So there are 2 types of cell division. One is mitosis, and the second one is meiosis. So the 2 types of cell divisions are mitosis and meiosis. So let's look at mitosis. Mitosis is a type of cell division that takes place in the body cells. Please mute your microphone so that others will hear you, so that others can hear. Just mute your microphone. So we are going to look at mitosis. I said that mitosis is a type of cell division that takes place in the body cells or somatic cells. The type of cell division that takes place in the body cells or somatic cells, that is mitosis. Body cells like your hands, your legs, your eyes, your head, these are different types of parts of the body. So division that takes place in this your body that enable you to grow is known as mitosis. Now let's look at meiosis. So meiosis is a type of cell division that takes place in the sex cell. In the sex cell, the division that takes place in the sex cells is meiosis. What are the sex cells? The reproductive cells, the sperm, the ovary. These are the division that takes place in your sperm, that takes place to produce small sperms or to produce eggs, is known as what? Meiosis.
Meiosis.
So these are the 2 types of cell division, mitosis and what?
Meiosis.
Very good. Now, whether you are discussing mitosis or whether you are talking about meiosis, there are stages of cell division. There are stages of cell division. And these stages are represented by a memory chip known as IPMAT. IPMAT is a memory chip. IPMAT. IPMAT is a memory chip that helps us to remember stages of cell division. The question is, what is I? I is interface. I in that IPMAT is interface. P is prophase. P is prophase. P is prophase. M is metaphase. M is metaphase. A is anaphase. A is anaphase. And T is telophase. T is telophase. So we have been able to look at the stages that takes place in the cell division. I used IPMAT to indicate the stages of cell division.
Number one, interface.
So the first one is interface. The second one is, I said the first one is interface. The second one,
Metaphase.
yes. The second one is prophase. IPMAT, IPMAT, IPMAT. The first one is interface. The second one is prophase. The third one is metaphase. The fourth one is anaphase. And the fourth one is telophase.
Okay.
So these are the
Please
stages
come
of
again,
cell
please come again.
These are the stages of cell division. You must know these stages because it helps you to remember the stages of cell division. Now, so whether you are talking of mitosis or meiosis, these stages apply. These four stages apply, no matter what you are talking. Whether you are talking of prophase or interface, whether you are talking of mitosis or meiosis, these stages must apply. Though they are not separated, they occur simultaneously. These stages occur simultaneously. They are not like say interfaces appear at this hour and then prophase will appear at this part. No, they are simultaneously occurring, meaning that all of them are occurring at the same time. So, but we divided it into these stages for understanding sake. For purpose of understanding, we're able to separate the stages. So, but in the real sense, they are simultaneous. That means they occur simultaneously. So, having known this, let us look at the details of these cell divisions. Maybe details of mitosis. After looking at details of mitosis, then we'll look at details of meiosis. So that's what we're saying. We can look at these stages of cell division. After we look at mitosis, after mitosis, we look at meiosis.
Sir, it seems the lecture material you are using is different from the one we have.
So let's look at mitosis, the stages of mitosis. Let's use, for example, a cell that has 4 chromosomes. You may be asked to draw and label the stages of mitosis in a cell that has 4 chromosomes. So you will be able to draw the stages of this mitotic division. So let's assume the cell has 4 chromosomes. So the first stage we are going to look at is interphase. The interphase in that cell that has 4 chromosomes is so the chromosomes are compact that you may not be able to identify them. That is the interphase. It's not easy to identify the chromosomes. From interphase, we go to the prophase. The prophase are divided into 2, the early prophase and late prophase. At the early prophase, the chromosomes begin to contract, and then you'll be able to identify the chromosomes. And then the nuclear membrane will now begin to disintegrate, and the centroids begin to form. Then at late prophase, you find out that the nuclear membrane has disintegrated, and it is possible for you to note the number of chromosomes, in this case 4 chromosomes. So that is the interphase, and then the prophase that is made up of early prophase and late prophase. The next stage of this mitotic division
please come again with the prophase.
The metaphase. Also, we have early metaphase and late metaphase.
Oh.
At early metaphase, you find out that the nuclear membrane has disintegrated completely, and then the centroids have started to move to the opposite pole. The centroids have begun to move to the opposite pole and even reach the opposite pole. And then at late metaphase, the 4 chromosomes that which you are using as an example have come to stay at the equator of the cell. The term is that these chromosomes will align themselves at the equator of the cell, at the center of the cell, being held by the spindle fibers. So at late metaphase, this chromosome will align themselves at the center of the cell, being held by the spindle fibers. So the next phase after metaphase is what? The anaphase. What happened during anaphase? Remember that a chromosomes have daughter chromatids. So at anaphase, the daughter chromatids will begin to migrate, to separate. The daughter chromatids of a given chromosome will begin to separate. So these 4 chromosomes that have aligned themselves at the center of the cell at metaphase, the chromatids will begin to separate, half going up, migrating up, and half migrating down. If you look at the diagram in your text, you see it. So at late anaphase, they have migrated and reached the opposite poles. So the daughter chromatids of the 4 chromosomes will migrate and move to the opposite pole. Early anaphase, they are migrating. Late anaphase, they have reached the opposite pole. The next phase is the telophase. Telophase is division. A constriction will now begin to appear to show you that the cell is dividing. You have early telophase and late telophase. At the early telophase, you will see the constriction. At late telophase, the cell has completely divided, giving rise to 2 daughter cells. That is mitosis for us. That is the stages of mitotic division. So in mitosis, one cell divides to give rise to 2 daughter cells. And the 2 daughter cells that are given, that have formed, have the same number of chromosomes as the parent cell. That is very unique of mitosis. So in summary, in mitosis, after mitosis, one cell divides to give rise to 2 daughter cells. And each of the daughter cells has the same number of chromosomes as the parent cell. If you are through with this, then we can move to meiosis.
We are through with it, sir.
Now, in meiosis, I tell you that meiosis takes place in the sex cell. That means in the sperm. Meiosis takes place to give rise to sperm and to give rise to egg. Remember, this thing takes place maybe in human beings when you have reached the age of puberty, maybe the age of 12 for a man, and maybe 11 to 13 for a female. But females mature earlier than male. So it prepares you to be a mother. If meiosis did not take place in your body, you cannot be a mother, you cannot reproduce. So God has designed this, both male and female, so that he now divides the number of chromosomes in your body, and then the chromosome in the sperm or the chromosome in the egg will be half. So that when the half of the sperm meet the half of the egg, both of them fertilize, then both of them will now form, preferably, the complete chromosome. So let's look at the process of meiosis. The process of meiosis is like meiosis undergoes 2 stages of cell division. You have the first meiotic division and second meiotic division. So meiosis have 2 stages, unlike mitosis that have only 1 stage. Meiosis have 2 stages of cell division. Let's look at this first stage. The first stage is called first meiotic division. while the second stage is called second meiotic division. So the question is, what happens during the first meiotic division? Let's still use our typical example of a chromosome, of a cell that has 4 chromosomes.
Okay, sir.
I can now tell you that a cell contains 4 chromosomes. With the use of label diagram, describe the process of meiosis in that cell. That is the assignment I'm going to give you at the end of the lecture. So let's do it now. So our typical cell has 4 chromosomes. So let's describe the process of meiosis. Of course, we're going to start with first meiotic division. First meiotic division. It's the same thing.
Please, are you sharing screen? I cannot see anything. No. Are you sharing your screen?
Yes, I'm sharing the screen.
I cannot see anything.
We are not seeing you, sir.
Are you not seeing me?
Even me, I'm not
No, we are
I'm not seeing
it,
any
sir. We are not
screen. No.
Seeing the
Yes,
screen,
sir.
sir.
We are not seeing.
Wow.
We are seeing.
Are you seeing me now?
Yes, yes, sir.
Wow. Okay, let me do it such a way that you cannot see me, but some are seeing me.
Yeah, we are seeing it now.
Okay.
Both you and the screen.
you. We are seeing
Very good. Okay, let's continue. So let's do the first meiotic division.
Sir, but what you are showing is Zoom, not the where you are reading from. What it is showing now is Zoom. Zoom meeting.
You say you are seeing me.
Sir,
And we are seeing the screen, but what you are seeing is Zoom.
There's something that's displaying on the screen.
There's nothing.
Okay, don't worry. Let's first of all pick. When we understand, then we go from there. Oh, just cool down. We are going to do it very fine.
Okay.
You understand?
Yes, sir.
So now let's do the first meiotic division. The first meiotic division. The first meiotic division, we're using our cell that has 4 chromosomes. We are using our normal cell that has 4 chromosomes. What we say here is that the stages of this first meiotic division is the same thing with mitosis. During interphase, nothing is seen. During early prophase, the nuclear membrane will start to disintegrate.
People should stop making noise now. They are talking too much.
So,
Sir, please mute everybody. You know it is too much. Mute everybody, please, sir.
Sir, please mute everybody. Please mute everybody. No one should speak.
okay.
How are we going to
like
ask
children.
questions if we are being muted, please? How are we going to ask questions if we are being
When
muted?
it is time, he will unmute us.
Mute everybody.
When it's time, he will unmute us.
Mute us.
One person should answer, not
Mute
everybody.
Now, what happened is that the first meiotic division is very simple. It's just like the mitosis that we discussed. So here, the chromosomes, the prophase, at the prophase, the nuclear membrane will start to disintegrate. And then at the late prophase, the centroids will begin to form. And then you now see that the chromosomes are now compact. You begin to see the chromosomes. Then at the metaphase, the chromosomes will come and align themselves at the equator of the cell. So they will come and stay, the 4 chromosomes will come and stay at the equator of the cell. But then they are not staying single, just like in mitosis. What happens is that they will stay in phase. They will pair up. So no longer single chromosomes at the equator of the cell. The chromosomes will now pair up. They will be in a phase. And there is a reason why they are in pairing up. The reason why they are pairing up is that something wants to happen. There's something we call crossing over. One daughter chromatins will cross to the other daughter chromatins, while the other daughter chromatins will come down and cross to the other one. So genetic materials are shared. So that is one difference between mitosis and meiosis. That in metaphase, instead of one single chromosome staying at the equator of cell, what is staying here is paired chromosome in pairs. And that pairs are called homologous chromosomes. So homologous chromosomes will come and pair up at the center or equator of the cell at metaphase. Then at anaphase, they will now separate. the homologous chromosomes will now separate. And as they're separating, the full chromosome, the full complement of the chromosome, one will go up and one will come down. The other one will go up and the other one will come down. So full complements of chromosomes will separate at anaphase. Then at telophase 1, that is telophase 1, then there will be a constriction showing that division is taking place. And at the end, the full complement of the chromosome will separate.
Please, can you come again on telophase?
Telophase, I say in telophase, a
Please
constriction
mute
now
everybody.
takes
We
place,
can't hear you clearly. We can't hear
showing
you
you that
clearly. Please mute
division is
everybody.
about taking place. And finally, they will separate. And when they separate, you now have two daughter chromosomes. But something happened. The chromatids of that chromosome have not yet separated. It is the entire complement of the chromosome that separates. So the chromatids of the chromosome must separate. And that is why each of the daughter cells formed will undergo second meiotic division. Each of the daughter cells will now undergo the second meiotic division. Of course, you know that in mitosis, there is no second meiotic division, but in meiosis, there is second meiotic division. So each of the daughter cells formed will now undergo second meiotic division. And what happened here? They will now undergo second meiotic division. So let's see what happened during second meiotic division. Now, during second meiotic division, we now have prophase 2, metaphase 2, anaphase 2, and telophase 2. So let's see what happened there. Let's start with the prophase. In prophase 2,
We can't see what you are showing us, sir.
in prophase 2, I will give you, don't worry, I will upload everything for you. But just understand the process. Now, at prophase 2, the chromosomes become very compact. And then the centroid, the nuclear membrane.
Please mute everyone. Please mute everyone. Mute
Sorry, sir, can you come again?
I say at prophase 2, as usual, the nuclear membrane will begin to disintegrate. The centroids will begin to form and migrate to the opposite pole. And then at late prophase, the nuclear membrane has completely disintegrated. And then we now enter the metaphase 2. At metaphase 2, the chromosome will come and align themselves at the center of the cell. And when they align the center of the cell, then the daughter chromatids has to separate. So the daughter chromatids will now separate after aligning themselves at the center of the cell. And then we will now move to anaphase 2. At anaphase 2, the daughter chromatids will separate and migrate to the opposite pole. So they will now move to the opposite pole, and at telophase 2, constriction will take place and the cell will completely divide. And so the two daughter cells will now divide. The first one divide into 2. The second one will also undergo the process and divide into 2, so that in the end, 4 daughter cells are formed. In the end, 4 daughter cells are formed from one single cell. If you compare it with mitosis, at the end of mitosis, 2 daughter cells are formed. But at the end of meiosis, 4 daughter cells are formed. But these 4 daughter cells that are formed, each of them has half the number of chromosomes as the parent cell. Each of them has half the number of chromosomes as the parent cell. So the sperms that will be formed, if it is the sperm, they will all have 23 number of chromosomes instead of 46 number of chromosomes. The chromosomes will be completely divided into half. And the egg that is formed, assuming it undergoes meiosis, it will be what? 23 chromosomes in each egg instead of 46. Such that the individual now has now prepared itself. If it's a male, after the age of 13, the number of chromosomes in the sperms will be what? 23 each. If it's a female, after the age of 13, the number of chromosomes in the egg will be what? 23. So that at the age of 25 or 30, when you get married, then if the young man, your husband, produces a sperm, the sperm is releasing has 23 chromosomes. As a woman, when you release an egg, of course, after fertilization, remember that the sperm will now enter through the vagina to the what? To the uterus. And it stay in the uterus, waiting for the egg to be released. When the egg is released, the sperm that contains 23 chromosomes will now fertilize an egg that contains what? 23 chromosomes, so that a zygote will be formed. And this zygote contains complete number of chromosomes, which is 46. A young individual is born. So that young individual that is 1 day old will grow to become 1 month old, and grow to become 12 months old, and grow to become 2 years old, 3 years, 13 years old, and the process starts again. That is the procedure for meiosis.
Please, can you come again?
So the next thing we are going to look at is what are the importance
Hello, sir.
of
Sorry, sir. Can you come
meiosis?
again?
You mean the whole process again? Okay. I will do a recap, but
This
allow us
age.
to. I will do a recap, but allow us to just finish.
Okay.
Now, what are the importance of mitosis? What are the importance of meiosis? That is the next question that we may ask you. The first one is importance of mitosis. Why does mitosis have to take place? It has to. Mitosis is important because it helps an individual to grow. Without mitosis, you will not grow. You will become just like that very day you were born. You will not be as big as you are now. So mitosis helps in the growth of what? The body cells. There is a continuous growth until you reach a full-fledged human being. In plants also, there is continuous growth even throughout the life of an organism, of the plant. So mitosis helps in the growth of what? The individual. If you have a cut, if you have an accident, and you go to the hospital, maybe to orthopedic surgeon, after the surgery, it is mitosis that helps that place to heal. Otherwise, it will not heal. So mitosis is necessary for the growth and heal of the body. So the second one, it helps in the multiplication of cells. Mitosis helps in the multiplication of cells, especially in lower organisms, bacteria and microorganisms. They divide very fast. So it is this process of mitosis that enables them to divide. If you have a cassava stem and you plant this cassava stem, it is mitosis that enables that cassava stem to what? To grow. So another one is that mitosis helps us to produce individual that has the same genetic makeup as the parent. It helps to produce individual that has the same genetic makeup as the parent. So these are some of the advantages of mitosis. Some of the importance of mitosis. We can also look at the importance of meiosis. Why are meiosis very important?
Please, number 3, you say it helps to produce individual that has
with the same genetic makeup as the parent. Do you understand?
Yes, sir. Thank you so much.
Do you understand?
Yes.
Okay.
Yes, sir.
Now, can we look at the importance of meiosis? Meiosis is important because you need to reproduce. Organisms need to produce young ones. If meiosis did not take place, you cannot have a child. You will just remain like that. So it is important for reproduction, for you to have young ones that will replace you when you die. When you marry, you need children in your house. So this is meiosis helps to reproduction. Then it also helps to produce individuals that have what? Mixed genetic makeup. If you marry a tall husband and you are a short woman, the tendency is that you have children that have what? Middle height. There was a genetic reshuffling. If you are not very intelligent and you marry a man that is very, very intelligent, the probability that your children will have what? You produce children that have mixed genetic makeup. Some of them are intelligent. If you are very, very black and you marry a wife that is very, very fair, the possibilities that to produce children that are fair in complexion. So these are some of the importance of what? Mitosis and importance of meiosis. But are there differences between mitosis and meiosis? Yes. So let's look at the differences between mitosis and meiosis. 1. Mitosis takes place in the body cells. Mitosis takes place in the body cells, while meiosis takes place in the what? Sex cell. Mitosis takes place in the body cells, while meiosis takes place in the sex cell. 2. Mitosis involves only one stage of division, while meiosis involves 2 stages of cell division. 3. There is no crossing over under mitosis.
So, okay, let's begin with the number 1.
There is no crossing over under mitosis, but there is crossing over under meiosis. There's a genetic mix-up under meiosis.
Yeah, please repeat number 2.
Number 2. Number 2, I say mitosis involves only one stage of cell division, but meiosis involves 2 stages of cell division. Then number 3, there is no crossing over. There is no crossing over under mitosis, but there is crossing over under meiosis. Then number 4, homologous chromosomes do not pay up. Homologous chromosomes do not pay up under mitosis, but under meiosis, homologous chromosomes pay up.
Sir, please, you are rushing.
Sorry, sir, what do you mean by they don't pay up?
Now, remember that we said that in mitosis, it's only one single chromosome that stays at the center of the cell during metaphase. But in meiosis, they go in phase. They stay in phase, of which one hand of the other one will cross over to the other side. One hand of the other one will cross, and then they will mix up. So that's why we say homologous chromosomes do not pay up under mitosis. The homologous chromosomes, they pay up under meiosis. Are you following? Now, under mitosis,
we are not following.
Under mitosis, 2 daughter cells are formed. Only 2 daughter cells are formed under mitosis. But under meiosis, 4 daughter cells are formed. Under meiosis, 4 daughter cells are formed. Then finally, under mitosis, each of the 2 daughter cells formed have the same number of chromosomes as the parent cell. Each of the 2 daughter cells formed have the same number of chromosomes as the parent cell. But under meiosis, each of the 2 daughter cells formed have half the number of chromosomes as the parent cell. Each of the 4 daughter cells formed has half the number of chromosomes as the parent cell. So these are some of the differences. I won't give you all. You do the assignment and get all. Now, we go to the next level. Let me summarize. So let's go to the next
Who
one.
are these heartless people now?
The next one. Okay, can we continue? The next one is the cell cycle.
Do us a favor of muting people's mics now. We are not hearing you.
So let's go to cell cycle. Cell cycle.
I have a question, sir. I have a question.
Cell cycle means series of events that takes place in a cell as it grows and divides. The events that takes place in a cell as that cell grows and divides. So that is the cell cycle. The exam, I will say, define the term cell cycle.
Sir, come again, please.
Cell cycle. What is cell cycle? define the term cell cycle. That's what I'm going to ask you in the exam. And I say that cell cycle. The cell cycle, they are events that takes place in a cell as the cell grows and divides. Events that takes place in a cell as that cell grows and divides. That is the cell cycle.
As the cell grows and what?
Divides. Now, you can look at stages of the cell cycle. Stages of the cell cycle. There are 5 stages of the cell cycle. In the exam, I will say list the stages of the cell cycle.
Number 1.
List 5 stages of the cell cycle. That's what I'm going to ask you in the exam.
Hello, sir.
So let's look at the stages of cell cycle. Stages of cell cycle. 1 is, if I were you, there are 5 boxes. I draw 5 boxes. These 5 boxes represent the stages of cell cycle. So draw 5 boxes. The first box, the second box, the third box, 4th box, and the 5th box. Then we'll label the boxes.
1, 2, 3, 4, 5.
Yes, sir. Label the first box G1. G1. Capital G1.
Done.
So we call it G1 phase. G1 phase. Then label the first G1 phase. Then the second one. What did you say? The first one is S phase. S phase. S, capital S. S phase.
Sir, S as in snake, right?
Sir, the first one.
S, yes.
First one.
S phase. S in bracket, synthesis phase. S in bracket, synthesis. S-Y-T-H, S-Y-N-T-H-E-S-I-S. Synthesis stage, known as S stage. That's the second box. Have you done that?
Yes, sir.
Yes, sir.
The third box, the third box is called G2 phase. The G2 phase, that is G means gap, G-A-P, gap. G2 in bracket, gap phase. G means gap. You have 2 G's, G1 and what? G2. The first one is G1 or G1 stroke gap 1. Then you have S phase. Then you have G2 or G2 in bracket gap phase.
So is that in stage? Is that in stage 3?
Yes. G1 is a phase. S is a phase. G2 is a phase. Have you written the 3 in the box you have drawn?
Yes, sir.
On top of the box.
Then these 3 boxes, that means G1, the S phase or synthetic phase, and G2 phase, they all occur during the interphase. And so somebody can bracket all of them and say occur during interphase. Occur during interphase.
We love mobile.
Occur during interphase.
Occur during interphase.
Yes.
But the question is, do you know what happened during interphase? Interphase is a resting phase. It's a resting phase. But actually, the cell is not resting. The only state is for us to remember that this is a state that many things happen. You understand?
Yes, sir.
So can we go to the 4th box?
Yes.
Can we go to the 4th box?
Yes, sir. Go ahead, sir.
The 4th one is called, label the 4th one, write it mitosis. Mitosis.
Okay, sir.
And then the last box. Last box.
Yes, sir.
Label it Cytokinesis. Kinesis.
Is it
Now,
5 boxes or
Is that a
6?
KY or a KI?
Is a KI.
Cyto,
It's K. It's KI.
CYTOKINES.
KINES.
Cytokinase.
OIS.
Yes.
Sir,
Now,
is it 5 boxes or 6?
this mitosis and cytokinesis, both of them occur during M phase. Both of them occur during M phase. M phase. While the other one, G1, S, and G2 occur during what?
Interface.
Interface, yes.
Yes.
Can you spell the M, please?
M, capital M dash.
Capital M, then face.
Yes. Now, can we now explain what happened at each phase? Is it possible for us?
Yes, sir.
Okay.
Yes,
Which
Now,
is the
let's look
phase?
at G1 phase. G1 bracket gap phase 1. G1 bracket gap phase 1. So if I ask you to list the phase of a cell cycle, then we are going to remember G1, S
phase,
mitosis,
G2
and
phase,
what?
mitosis and
Cytokinesis.
cytokinesis.
Can you do that?
Yes, sir.
yes, sir.
Okay, so let's see what happened during G1.
Sorry, please, can you repeat that?
Let's see what, there is no time, so I have to be fast. Let's see what happened during G1.
No time.
So what happened is G1 is that it is the growth phase of a cell. The growth phase of the cell.
It is the
growth phase of the cell.
phase of the cell.
Yes. Do you understand? The
Yes,
growth
sir.
phase of the cell.
Yes, sir.
So, and it is the longest phase. It is the longest phase.
It is
The longest phase. Yes.
longest phase.
Yeah, the second one, S phase or synthetic phase. Synthesis phase. Synthesis phase is this phase
Please come again about
in which the cell synthesizes a replica of DNA genome.
In which the cell
synthesizes a replica of its DNA. Synthesizes a replica of DNA.
of DNA.
That is DNA replication.
Okay, sir.
Then G2 phase. G2 or G2 gap.
Can you come again with that second one?
So yes, it is the stage of the beginning of mitosis.
It's
Stage of the beginning of mitosis.
of the beginning of mitosis.
Yes. The cells prepares. The cells prepare itself.
The cell?
Division. The cell prepare itself for division. The cell prepare itself for division.
Okay, sir.
Now,
In conclusion, G1, S, and G2 phase constitute the interphase.
Yes.
In conclusion, G1, S, and G2 phase constitute the interphase.
Yes, sir.
Now let's go to the M phase. The M phase. And you know that the M phase is made up of what? Mitosis and what? Cytokinesis.
Yes.
So let's look at mitosis. Mitosis.
Okay, sir.
It is the stage in which two cells are formed at the end. This is the stage in which two cells are formed at the end.
At the end.
Yes, two cells are formed at the end of mitosis.
Of mitosis.
Yes, and the stage is subdivided into 5. The mitosis is subdivided into 5.
Into
The
5.
interphase,
Yes.
the prophase, the interphase, the prophase, the metaphase, anaphase, and what? And what?
Telophase.
Telophase. In bracket, IPMAT, IPMAT. Everything in capitals. IPMAT. Is that okay?
Yes, sir.
Then finally, cytokinesis. Cytokinesis. This is where the cytoplasm divides. The phase of the cell where the cytoplasm divides, creating two daughter cells. This is where the cytoplasm divides, creating two or more daughter cells. Creating two or more daughter cells. Are we okay?
That's your last statement, sir.
This is where the cytoplasm divides, creating two or more daughter cells. Are we okay?
Yes, we are.
Okay. Now let's move to a new one, a new title. A new title is viruses. Virus. Yes, we want to look at virus. So viruses are microorganisms that can be seen with only the use of electron microscope. They are microorganisms that can only be detected or seen by the use of electron microscope. They are very tiny, tinier than even bacteria. Bacteria can be seen with the use of optical microscope. Bacteria can be seen with the use of optical microscope. Bacteria can be seen with the use of optical microscope, but virus cannot be seen with optical microscope. Can only be seen with the use of electron microscope. Very tiny, very minute.
Yes, sir.
So, at the end of our study of viruses, we'll be able to draw a generalized virus. We'll be able to draw the structure of a, just listen. I said at the end of our discussion of these viruses, we'll be able to draw the structure of a virus.
Okay, sir.
We'll be able to discuss the virus as an agent of disease, diseases that are caused by virus. We also look at the characteristics of virus, because they have one kind of character. They behave in a very special way. So we look at characteristics of virus. And then we draw a life cycle of retrovirus and HIV. We draw a life cycle of retrovirus and HIV. And then we look at some treatments. We list some treatments of HIV/AIDS. These are what we are going to do when we finish the discussion of virus. After the discussion of virus, we pick another one, fungi.
Okay,
So let's just finish it. What are the characteristics of a virus? What are the characteristics of viruses? Viruses cannot be seen with our naked eyes. As we said, we can't even use optical microscope to see them. We cannot be seen with our naked eyes. It cannot even be seen with the use of optical microscope. They can only be seen with the use of what? Electron microscope. They are very simple, but they do not have cell structure. Even though they are simple, but they don't have cell structure. So that's why their behavior is very, very interesting. They don't have cell structure.
So please, can you come again?
And then they live inside an organism. So they are parasites. They live inside an organism. And so they are parasites. And remember that parasites are organisms that live in or on the body of other organisms and derive nutrients from that organism. They may even cause harm or even death to the host. So, if we look at the structure of a virus, find out that the virus have DNA inside or RNA inside, and then they have a protein coat that cover it. That's how they look. The DNA, that's the RNA or DNA is inside, and they have a protein coat that cover it. That's just the simple structure of a virus. They are very simple, but they behave in a very unfriendly manner. So let's look at what happened, the life cycle of erythrovirus or HIV. So how does it behave? What is the life cycle? Now, especially, you know that it lives in the host is man, human being. So what happens is that when it enters your body, it moves into your cell and attaches itself to the receptor protein in the cell membrane. When it comes into the cell of your body, it attaches itself in the receptor protein of the cell membrane of your cell. Remember, all these things cannot be seen with our naked eyes. But the first time we discussed cell, I told you that cell is the building block. It's the smallest structure of the building structure of a human being, of an organism, the building block of an organism, the structural and functional unit of life. So when this retrovirus or virus enter your body, then it goes to your cell membrane and attaches itself into what? A special what? Receptor protein. So, and then enter into your body by endocytosis. Endocytosis, it now enter and moves inside your main cell. And when it enters into your body, the viral RNA in that virus will enter into the cytoplasm of your cell, together with an enzyme called what? Transcriptase, reverse transcriptase. They will enter the cytoplasm. So the RNA, viral RNA, the RNA of that virus will now enter into your cytoplasm with the aid of another enzyme called reverse transcriptase. And then that RNA is now transformed into what? DNA, double-stranded DNA. So from one single RNA of the virus, it now transmutases into a double-stranded DNA. And then their next target is to move into the nucleus of the cell. And when they enter into the nucleus of the cell, they now do what? Insert itself into the host DNA. Once they enter the nucleus of your cell, they now put themselves into your DNA present in the nucleus, in your own nucleus. From there, it divides and makes another virus DNA. They will continue to divide. Anytime the cell divides, a new DNA, a new viral DNA is formed. From then, it now replicates and then become plenty in your body, to the extent that it will now start killing the cells of your body, and the cells will continue to collapse and die. Before you know, it takes over the entire cell. And then it graduates and becomes the AIDS. So AIDS, enter into the AIDS. No longer HIV, now enter into this AIDS. But luckily, there are some stages, there are some recommendations or drugs that can help us to take care of that. So after about 5 years, the whole thing divides so much that it's not overpower your immune system, and you are down with AIDS. So that is just the simple way that this virus enters our body. It enters and then takes possession of our body, and then compromise our immune system will now be compromised. So let us do a summary of what we did today, because I will use the remaining 5 minutes and do a summary of what we have discussed today. So let's do a kind of summary of what we did today. So today we look at cell division.
Yes, sir.
And we said that cell division is division that takes place in the cell. Whether you are talking of plant cell or whether you're talking of animal cell, division must take place for the animal to grow and for the plant to do what? Grow. And so we have two types of cell division: mitosis and what?
and
Meiosis.
meiosis.
Mitosis takes place under body cells or somatic cells. On your head, in your eye, in your ear, in your mouth, in your body, in your legs, every part of your body, for you to grow and become a full-fledged adult. Even when you're an adult, if you have a cut, mitosis will help you to heal that place. And meiosis is the type of cell division that takes place in what? The reproductive cells, the sperms, and what? The eggs. They are called sex cells. And why do they take place? Because it will go, why meiosis takes place is that at the end of meiosis, the number of chromosomes in the sex cell will be reduced to half. So that the sperm now has 23 chromosomes and the egg now has 23 chromosomes. So that when they unite later in life, after marriage, the two join again and form 46 number of chromosomes as a new individual in a zygote. Now, we say whether we are talking of mitosis or meiosis, there are 5 stages of cell division. And we use ip math to represent it. And ip math is interphase, then prophase, then metaphase, anaphase, and what?
And telophase.
And these are 5 stages. Then we say, let us start with mitosis. We use a typical cell that has 4 chromosomes as a demonstration. We say under mitosis, when this cell undergo mitosis, it passes through interphase, prophase, which has early prophase and late prophase, passes through metaphase, early metaphase and late metaphase, and then it passes through anaphase, early anaphase and late anaphase. It passes through telophase, early telophase and late telophase. And in the end, 2 daughter cells are formed. And these 2 daughter cells that are formed have the same chromosome as the parent cell.
Yes, sir.
Now, we now go to meiosis. We say meiosis is type of cell division that takes place in the reproductive cell. And at the end of meiosis, now the meiosis has 2 stages: first meiotic division and second meiotic division.
Yes, sir.
First meiotic division, if you are using our example of a cell that has 4 chromosomes, during the metaphase, homologous chromosomes will pair up at the center of the cell. They stay in pairs. Unlike in mitosis, where they stay 1-1. In meiosis, first meiotic division, they stay in pairs for crossing over to take place. So after everything, the cell will now divide into 2. And the 2 cells will now undergo second meiotic division. And after the second meiotic division, each of the cells now divide into 2. So the total number of cells formed are 4. And if you look closely, each of the 4 cells formed have the number of chromosomes as the parent cell. This is very, very, very interesting. So what helps us is that it helps meiosis helps in the reduction of the cells, so that you now have the number of chromosomes. The time is almost going. You have 3 minutes more. Then we look at advantages of mitosis, which helps us to what? Grow. Then it helps also in the multiplication of cells, and it helps to produce exact copy of the same chromosome. Then we look at the importance of meiosis. We say it helps in reproduction. It helps us to mix up our genetic material. Then we look at differences between mitosis and meiosis. We say mitosis takes place in the body cells, meiosis takes place in the sex cells. Meiosis involves mitosis involves there is no crossing over in mitosis. There is crossing over in meiosis. Homologous chromosomes do not pay off. The homologous chromosomes pay off in meiosis. Then we say 2 daughter cells are formed under mitosis, but 4 daughter cells are formed under meiosis. Then we now enter the cell cycle. We say that cell cycle are a series of events that takes place in a cell as it grows and divides. And then the cell cycle involves 5 stages. You have the G1 phase, you have the S phase, you have the G2 phase. G1, S, and G2 make up the interphase. Okay, then you have the what? The M phase. And the M phase involves mitosis and cytokinesis. virus, and then you look at as you can only see virus with the use of electron microscope, and then we look at characteristics of virus, and also we look at the life cycle of the HIV virus. This is where we call it a day, and I want you to do some exercise. Use a cell that has 4 chromosomes. With the use of label diagram, describe the process of mitosis and meiosis. And then B part of it is list 10 differences between mitosis and meiosis. These are the assignments we are going to do.
Please come again. Oh.
Can you say it again, sir?
A cell contains 4 chromosomes. With the use of label diagram, describe the process of mitosis and the process of meiosis.
For one cell.
1A. Then the B part of it is list 10 differences between mitosis and meiosis. And then C part of it, the C part of it is list the 5 advantages of mitosis and 5 advantages of meiosis. This is your assignment. And this is why we call it today. I thank you very much for participating today.
Please, I didn't get the assignment.
The advantages and disadvantages.
Sorry, sir, can you drop the PowerPoint for the lecture?
How many are the advantages and disadvantages?
Yes, we are going to. I'm going.
5 advantages of mitosis and 5 advantages
that you can look at
If you get the assignment, can you go to the
Thank you, sir.
So thank you. That's why we call it.
Let's go to the group. Send the assignment to the group.
Who submitted the assignment?
So how we do it?
Hello, sir.
Yes, submit as usual. Thank you and God bless.